Chapter 7 is all about how we are harvesting energy from the food that we eat. This is a cellular process. So where is the energy in food? Well, the energy for life is obtained by breaking down the organic molecules that we ingest that are originally produced in plants. And the energy from those organic molecules is made available to us by stripping away the electrons and using them to make ATP, our energy molecule, adenosine triphosphate. This whole process that we're talking about is called cellular respiration. So whenever you hear cellular respiration, think to yourself, using glucose to make energy. Eukaryotes produce the majority of their ATP by harvesting electrons from the food molecule glucose. You can see the chemical reaction for this process shown here. You can see we've got glucose as our first reactant and oxygen as the other reactant. And then our products are carbon dioxide, water, and here, energy, the main goal of this process. So while our cells are trying to get this energy, and that's the goal of the whole process, we do have byproducts of water and carbon dioxide. And when you kind of zoom out of just looking at the reaction and think about how we function, it makes sense because we eat food, so here's our glucose, we breathe in oxygen, we breathe out CO2, and we will always have water in our system. So this is a logical byproduct of this process as well. This is kind of a basic overview of the 3 steps of cellular respiration. I wanted to start out simple and then get more in depth. So it's good to keep in mind we've got 3 steps: glycolysis, and then the citric acid cycle, which is also known as the Krebs cycle, and the electron transport chain. Before we go any further, I do want to point out that a lot of times in biology and science in general, there are multiple terms that describe the same thing or the same process. Usually there's a preferred term among scientists and then some extras that are also acceptable. But in this case, pretty much any biologist or scientist that you talk to could either use the Krebs cycle or citric acid cycle. They are both very commonly used interchangeably. So if you could just kind of think of it in your mind as the Krebs cycle / citric acid cycle, that's going to help you prevent confusion in the future, because you really want to know those are the same thing. And anytime you hear either of them, we're talking about the same thing. Another thing I want to point out while we're here is these 3 steps are not all happening in the same part of the cell. So our first step, glycolysis, is actually out here in the cytoplasm. And then we enter the mitochondria. That's where we will begin our Krebs cycle or citric acid cycle. And we're still in the mitochondria for the 3rd step of cellular respiration, which is the electron transport chain. When we look at how much ATP is produced in each part of this process, you can see there are only 2 molecules of ATP in glycolysis. There are 2 molecules of ATP produced in the citric acid cycle. And then there's a whopping 32 molecules of ATP coming from the electron transport chain. So really the heavy hitter here that's providing the most energy from this process is the electron transport chain. But it couldn't function without the processes of the citric acid cycle and the products of glycolysis. So they're all working together. Our glucose is transformed into energy through these 3 steps. The main goal of glycolysis is to basically make glucose into a molecule that can enter the mitochondria for the next step. And that's what it does. Glycolysis breaks down glucose into a molecule called pyruvate, which is a word that you need to remember. So we need to know in glycolysis, our goal is to break down glucose into pyruvate. After we've done that, pyruvate is going to head to the mitochondria to kick off the next steps. As it enters the mitochondria, it will be transformed into acetyl-CoA, another word that we need to remember. So pyruvate becomes acetyl-CoA as it enters the mitochondria, and that is what is going to kick off the citric acid cycle and lead to the next step, the electron transport chain, where the majority of our ATP is made. So more details on our first step, glycolysis. In glycolysis, like we said, we're breaking down glucose to form pyruvate. This is happening in the cytoplasm of the cell, and it's also important to remember it's an anaerobic process. So there's not a lot of available oxygen just floating around in the cytoplasm. And that might be something that helps you remember which part of the process does not need oxygen, because it's this first step. The other two steps both utilize oxygen, or in an oxygen-rich environment. There are 2 molecules of ATP produced for every pyruvate molecule that is made. But the main goal of this step is not producing ATP. The main goal is producing this pyruvate so that we can go into the mitochondria and kick off the citric acid cycle. Pyruvate molecule is going to be converted to acetyl-CoA during that transition step as the pyruvate is entering the mitochondria, so that it can be used in the Krebs cycle. Once the Krebs cycle has initiated, we are harvesting energy from that acetyl-CoA. Remember, acetyl-CoA used to be pyruvate, pyruvate used to be glucose. It's just been made usable. This is taking place in the inner membrane of the mitochondria, and it is an aerobic process, so oxygen is needed. Aerobic. Oxygen is needed. If this were anaerobic, like glycolysis, that would mean oxygen is not needed. But this is aerobic, so we are in an oxygen environment. There are also 2 molecules of ATP produced in this step, but really, the really good, useful thing about this step is how much NADH and FADH2 are produced. Now, we haven't encountered FADH2 before, but it's basically serving the same purpose as our NADH. We know from our last chapter, NADH functions as an electron donor. So it's a molecule that's often used in our cellular processes whenever an electron needs to be transferred for some kind of chemical reaction to proceed. So that's the main goal, because we need to provide the electron transport chain with electrons. And those are coming from the NADH and FADH2 that are produced in the other steps of this process. Once we get to the electron transport chain, this is where we are transferring electrons with redox reactions. And that's what's providing our heavy amount of ATP. Redox reactions are something that we're going to be introducing right now, so don't feel hesitant if you don't recognize that, but I've got a video that's really going to clear up what redox reactions are that'll hopefully make it stick for the long term for you. But those redox reactions are happening so that our transfer of electrons can take place, which is going to result in our ATP production. We're still in the mitochondria for this process, which means it is an aerobic process where oxygen is needed. In the electron transport chain, we're going to receive that NADH and FADH2 from the Krebs cycle. And you don't need to remember these numbers, but I just wanted to put them here to kind of keep in mind. There are about 10 molecules of ATP made for every NADH that's provided, and about 2 molecules of ATP are made for every FADH2. So our electrons are passing from atoms or molecules in many chemical reactions, including the ones that are involved in cellular respiration. So when we talk about redox reactions, that's really just the combination of these 2 kinds of reactions. So first of which is our oxidation reactions. And this is when an atom or molecule loses an electron. So if you're going through a reaction and the reactant molecule has lost an electron, that's an oxidation reaction. Reduction reactions are the opposite, when an atom or molecule has gained an electron. And this can be kind of hard for people to remember because it seems a little counterintuitive. But if we think about it, electrons are negatively charged. So if we are gaining an electron, that's a reduction of charge. We've lowered the charge because we gained something negative. And that is what helps it stick for me. So these reactions are always happening together and feeding into each other. They are cyclical. And we summarize them as redox reactions. We just took the first part of reduction and the first part of oxidation. In many redox reactions, oxygen is going to be a common electron acceptor. In cellular respiration, it's actually the final electron acceptor, meaning at the very end of the electron transport chain, we need something to accept the leftover electrons, and that molecule is going to be oxygen. Energy is going to follow the electrons, so the reduced form of an organic molecule is going to have a higher level of energy than the oxidized form. Here we can see we have lost an electron in this oxidation reaction. Our resulting molecule has a positive charge because we lost an electron. Here you can see we have gained an electron in this reduction reaction, ending up with a negative charge. And when we're saying it's a higher level of energy, we're also meaning it's more reactive, which we understand because it has this pull of the negative charge. So in glycolysis and the Krebs cycle, quick summary before we go further. Remember, glycolysis is in the cytoplasm. It does not require any oxygen to make ATP, and it's a very ancient pathway for lots of organisms. The Krebs cycle occurs within the mitochondria. It harvests energy-rich electrons through oxidation reactions, and the electrons are going to be passed to the electron transport chain in order to power ATP production. When we're passing those electrons to the electron transport chain, that's when we're passing on our NADH and FADH2. That's what's carrying the electrons to be donated. So this is a really great picture to help you memorize the flow of this and what's being produced at each step of the process and where each step is happening. Let's go through it together. So first we'll notice in both animal cells and plant cells, there are mitochondria. So plants use their chloroplasts to make sugars, and they also use the mitochondria to break sugars down for energy. That's like what happens whenever a seed is starting to sprout its first leaf and there's no other leaves because the plant hasn't grown enough. So plants use both processes of photosynthesis and cellular respiration. We don't have any chloroplasts and we can't photosynthesize, but we do have mitochondria, and that's how we're breaking down our glucose to get energy. So we're starting in the cytoplasm where there is no oxygen available to the process. And the goal of glycolysis is to transform glucose into pyruvate so that it can enter the mitochondria. You can see we're going to be making a little bit of NADH at each part of this process. So we're making a little bit of NADH at glycolysis, during the pyruvate oxidation, during the Krebs cycle, and we're also making that FADH2 in the Krebs cycle. All of those are going to feed into the electron transport chain as electron donors. So we've made glucose into pyruvate. Pyruvate is entering the mitochondria. As that happens, it will be oxidized into acetyl-CoA. This is the molecule that really kicks off the Krebs cycle. Once we've got our acetyl-CoA, you can see we will have our byproduct from pyruvate oxidation and from the Krebs cycle of CO2. So here our CO2 is leaving this process, and it's going to exit the cell, enter the bloodstream, make it back to the lungs, and be exhaled by the organism. Here's our little bit of ATP production that's happening in the Krebs cycle. Remember, 2 molecules here, 2 molecules here. And then the main goal of the Krebs cycle are electron donors, FADH2 and NADH. Once all of those electron donors have been supplied to the electron transport chain, we will have our byproduct of water. Remember that oxygen is our final electron acceptor, and really our electrons are being carried by our hydrogen atoms. So we have these hydrogen ions with available electrons, and the leftovers are going to be donated to oxygen, and that's where our byproduct of water comes from. Here's our main goal with this, which is the big amount of ATP that's produced in the electron transport chain. And then we will also have byproducts of NAD plus and FAD. If you could just think for a second, what do you think those are going to be useful for? Well, if you look at them, this is NAD plus, meaning it has a positive charge. And we used to have NADH with a neutral charge. We lost electrons. So this NAD plus is going to be recycled back into this process and become NADH again and bring more electrons. Same with FAD. So basically, these are just these molecules with the hydrogens and electrons associated with the hydrogens stripped away. They won't be thrown out. They won't be seen as waste. They will be recycled into the process to become electron donors again. Glycolysis is a sequence of reactions that form a biochemical pathway. And we talked about that in the last chapter. That's just basically the concept of we've got a reactant interacting with an enzyme that makes a product, and that product that was just made now becomes the reactant for the next reaction with a new enzyme to make a new product. And then that product becomes the next reaction, and so on, until we end up with a final product. But glycolysis is one of those biochemical pathways that is changing one thing into the next usable thing, into the next usable thing, until we end up with our final product. There are actually 10 enzyme catalyzed reactions that are happening. The 6-carbon sugar glucose is going to be broken into 3-carbon pyruvate molecules. And the breaking of the bonds is what yields that little bit of energy that is used to phosphorylate ADP into ATP. Remember, adenosine diphosphate is ADP because we've got 2 phosphates. And whenever it's phosphorylated, we've added a phosphate, so it becomes adenosine triphosphate with 3 phosphates. And that process is called substrate level phosphorylation. In addition, electrons and hydrogens are going to be donated to NAD+, so we have that little bit of NADH that came from the glycolysis process as well. So don't let this slide intimidate you because it's a pretty good simplification of what's happening in glycolysis. We start off with this 6-carbon glucose. Yes, the molecule is more complex than this 6-carbon chain, but that's a good way to simplify it when we're trying to learn what's happening here. So we start off with our 6-carbon glucose. It's going to become these 2 separate 3-carbon sugar phosphates. So now each molecule has 3 carbons and a phosphate. Those 3-carbon phosphates are going to donate a hydrogen to NAD+ for NADH production. And also we're going to remove a phosphate group, which is going to allow ADP to become ATP, and that's where our little bit of energy is produced in glycolysis. But here's the main goal, our 2 3-carbon pyruvate molecules. You can see they're 3-carbon sugar phosphates when they still have the phosphate. But once we break it off for that little bit of ATP, that's when they become our usable 3-carbon pyruvate. Whenever oxygen is available, the cell will harvest pyruvate's energy in 2 steps. So the first is we oxidize pyruvate to form acetyl-CoA. And that's the transition step between glycolysis and the Krebs cycle. And next we need to oxidize acetyl-CoA in the Krebs cycle. Whenever pyruvate is oxidized, that's when a single one of its 3 carbons is going to be cleaved off by the enzyme pyruvate dehydrogenase. That carbon is going to leave as part of our CO2 molecule, which is kind of a byproduct of this process. So the carbon is donated to this oxygen. So we've got CO2 leaving. In addition, a hydrogen and electrons are removed from pyruvate and donated to NAD+. So we take off a hydrogen from our molecule, give it to NAD+, and get NADH. Now our remaining 2 carbon fragments of pyruvate will be joined to a cofactor, which is usually, you could just think of it like a helper to enzymes, called coenzyme A. And that final compound is going to be what we're calling acetyl-CoA. So you can see that happening here as well. Glycolysis takes that 6-carbon sugar, turns it into a 3-carbon pyruvate. We break off one of those carbons and give off a little bit of CO2. We also get rid of a hydrogen and give it to NAD+. Coenzyme enters the reaction and combines with our 2-carbon molecule, and that's what gives us acetyl-CoA. So that's what we need to kick off the Krebs cycle. If we need energy, the acetyl-CoA will go into the Krebs cycle so we can be making ATP. If our cells are already flooded with ATP and we have more than we can use right now and we don't need any more, then instead we're going to switch into a process of fat synthesis. And acetyl-CoA is going to follow a different pathway because we're not trying to make energy, we have plenty of it. But we might as well make some fat that's energy we can use later. So that's what's happening. We're either going to go to ATP production and enter the Krebs cycle / citric acid cycle, or if we've got plenty of ATP, it's going to switch into fat synthesis so we can make energy to use another day or another time. This is really helpful because whenever we're transferring these electrons, really what we're talking about is transferring the hydrogen atoms. And this is a useful way to kind of visualize that. So here you can see we've got an enzyme that a substrate is intending to land. So the substrate is coming to the active site of the enzyme. There's also this other binding site for NAD+. Once NAD+ and our substrate have both bound to the enzyme, now they're held really close together and it's really easy for this electron transfer to happen. Also, I just wanted to point out, you can see that they showed a hydrogen, an available hydrogen atom attached to our substrate and an available electron. But really, this electron is part of the hydrogen. They're just trying to show you the value that the hydrogen is bringing, which is the electron. So once they're cozied up next to each other, we've got our substrate and our NAD+, which will be an electron acceptor. It's ready to accept an electron. It has a positive charge. The hydrogen is going to jump over to NAD+, so we will have a product of NADH and whatever the substrate has become since it just lost that hydrogen atom. So that's what's happening with our electron transfer. As we mentioned, the fate of acetyl-CoA is going to depend on how much ATP we already have in the cell. If there's not enough ATP, then acetyl-CoA is going into the Krebs cycle. If we have a whole bunch of ATP, then acetyl-CoA will be sent to fat synthesis so that we can store energy for another time. The Krebs cycle occurs in the mitochondria, and it's a series of 9 reactions that can be broken down into 3 stages of oxidative respiration because we're doing oxidation reactions. We don't really need to go as in detail as every specific part of the 9 reactions. We're trying to simplify this for introduction to biology, but these 3 stages are a good way to kind of break that down. So first stage is acetyl-CoA enters the cycle, and it's going to bind to a 4-carbon molecule, which forms a 6-carbon molecule. Because we know that acetyl-CoA still only has 2 carbons on it. It's going to come together with a 4-carbon molecule. Now we have a 6-carbon molecule. Next, 2 carbons are going to be cleaved off, and they're going to leave as CO2. So we've got even more of our CO2 byproduct now from the Krebs cycle. The electrons are going to be donated to NAD+ again, so there's a little bit of NADH production, and ATP will be produced. That's where we get our 2 molecules of ATP. The 4-carbon molecule is going to be recycled, and more electrons will be extracted, forming NADH and FADH2. Let's look at that happening here. We've got acetyl-CoA, a 2-carbon molecule, combining with a 4-carbon molecule to make our 6-carbon. This 6-carbon is going to donate an electron and carbon to have a byproduct of CO2 and a product of NADH. So there's some of our electron donor that's been made. And then that's going to happen again. So we just took off one carbon molecule. We went from 6 to 5. Now we're going to take off another one. Same thing. We donate that. It leaves as CO2. That extra hydrogen is donated to NAD+. It leaves as NADH and goes to the electron transport chain. We've got that little bit of ATP that's given off, and now a leftover 4-carbon molecule that's ready to be cycled right back in and combine again with another 6-carbon molecule. Or with another, 2-carbon molecule to become a 6-carbon molecule. And that just keeps going. So 2-carbon combines with 4. We get this 6-carbon molecule, get rid of a carbon and a hydrogen, get rid of a carbon and a hydrogen, give off ATP, recycle that leftover 4-carbon molecule, and so on. That's how we're providing our FADH and our NADH2 to the electron transport chain. In cellular respiration, the glucose is going to be completely consumed. So there's no part of the glucose that we're not breaking down and using. The energy from its chemical bonds will have been transformed into these things after glycolysis and the Krebs cycle. So we're not at the very end. We're just at the end of our 2 steps. At the end of glycolysis and the Krebs cycle, we will have made 4 molecules of ATP, about 10 NADH electron carriers, and 2 FADH2 electron carriers. Now that we've got those, we can start the electron transport chain. So NADH and FADH2 are going to come in and transfer their electrons to a series of molecules that are embedded in the mitochondrial membrane. That is the electron transport chain. Many of the proteins that are in the electron transport chain are going to operate as proton pumps. So they're pumping protons into the inner membrane space of the mitochondria. The very last transport protein will donate the leftover electrons to hydrogen and this says to hydrogen and oxygen, but really we're donating hydrogen to oxygen to form water. And that's how we get our byproduct of water. Mitochondria use chemiosmosis to make ATP. So we know that's the process when we're creating that proton gradient so that we can use the ATP synthase channel to turn ADP into ATP. That passage is what's powering the production of all of that ATP at the end of the electron transport chain. Let's look at that here. So brief overview. Glucose was turned into pyruvate in glycolysis. It was oxidized as it entered the mitochondria where there is oxygen. The Krebs cycle produced NADH and FADH2. All of the NADH and FADH2 made in every step of this process are fed to the electron transport chain in the mitochondrial membrane. And that's when we get our good flow of electrons through our transmembrane proteins. That's what's also going to provide this electron gradient, this concentration gradient of a bunch of hydrogen out here in the inner membrane space. Once it's all out here in the inner membrane space, now we've got a gradient that can force the ATP synthase channel to function. Whenever we're in the absence of oxygen, though, fermentation, rather than oxidation, is the pathway that's going to happen after glycolysis. So if there is no oxygen available, instead of going to the Krebs cycle, we're going to enter a fermentation process. In fermentation, an organic molecule accepts hydrogen atoms from the NADH generated by glycolysis and recycles NAD+, which allows glycolysis to continue. Bacteria carry out more than a dozen kinds of fermentation, and eukaryotic cells are also capable of fermentation, but only a few types. When we talk about ethanol fermentation, which happens in yeasts, NAD plus and ethanol are produced, and this is the source of ethanol for like wine and beer. There's also lactic acid fermentation, which happens in our muscle cells. And that is when NAD plus and lactic acid are produced. Both of those are fermentation processes happening in the absence of oxygen. So you can see the basic overview of that here. We don't need to go too in depth, but you can see we've got this cycle between NAD plus and NADH and a product of ethanol for yeast. Same thing here. We've got this cycle of NAD plus and NADH and a product of lactate, which is lactic acid here, happening in our muscle cells. But glucose isn't the only food molecule. We are also going to be getting energy from foods that are not just sugars. And proteins are first broken down into their individual amino acids that make them up. Deamination reactions are what convert the amino acids into molecules that can take part in the Krebs cycle. Also, fats are first broken down into fatty acids, which is a process called beta oxidation. This symbol is called beta. So beta oxidation converts the fatty acid tails into acetyl groups that can be combined with coenzyme A and can form acetyl-CoA, which is also needed for the Krebs cycle. So this is how we're utilizing those proteins and fatty acids. This overview is really useful to see what is feeding into this process. So you can see we've got our lipids and fats here, which will become fatty acids, and through beta oxidation can provide some acetyl-CoA for the Krebs cycle. The other thing that provides acetyl-CoA for the Krebs cycle is changing sugars into pyruvate and glycolysis. Another thing that can produce some pyruvate is whenever we deaminate those amino acids, that can result in pyruvate molecules that can then be transformed into acetyl-CoA as well. Other amino acids, other proteins that have been broken down into amino acids and then broken down further through deamination could become a waste product like ammonia. Same thing with nucleic acids that we're breaking down. They could either become products like a 4-carbon molecule that enter the Krebs cycle, or waste like ammonia. At the end of this process, after our pyruvate has become acetyl-CoA, we've produced NADH and FADH2, we've got our byproducts of water and carbon dioxide, and a bunch of ATP. So I think this is a great slide to help summarize everything we've talked about cellular respiration. We know that parts of the process are happening in the cytoplasm. That's the first step, glycolysis. And the other two steps, being the Krebs cycle and the electron transport chain, are happening within the mitochondria. So please remember, the cytoplasm does not have a lot of free-floating oxygen available. This is an anaerobic process that is happening in the cytoplasm. And as glucose is transformed into pyruvate, we're also going to give off a little bit of ATP and a little bit of NADH. As pyruvate enters the mitochondria, it goes through pyruvate oxidation. We will give off a little bit of CO2 from the carbons that have been cleaved, and a little bit of NADH electron donors there as well. Once that has happened, pyruvate has become acetyl-CoA. Acetyl-CoA kicks off the Krebs cycle. We'll have byproducts of CO2 again from the cleaved off carbons, a little bit of ATP, and then the main goal of the Krebs cycle are electron donors, FADH2 and NADH. All of the electron donors made in this process are going to the electron transport chain to support that electron gradient. So we'll have that gradient from our hydrogen ions that is going to allow for the ATP synthase channel to function. That synthase channel is going to be phosphorylating ADP into ATP with 3 phosphates. So that is where the majority of our energy production comes from. We'll also have a byproduct of H2O, which is from the oxygen accepting those leftover hydrogen. And we will also have these NAD plus and FAD that get recycled back in the process so they can become NADH and FADH2 again, and continue to support the electron transport chain. So, the processes in the cytoplasm are anaerobic, no oxygen needed. Within the mitochondria, they are aerobic. We do need oxygen. We need oxygen to accept the carbons, and we need oxygen to accept these hydrogens. I did want to go ahead and mention, though, we said in the beginning, we're producing about 2 ATP here, 2 ATP here, and about 32 ATP in the last step. But I wanted to point out that is not a concrete number. It can vary based on the organism, based on the time of day, based on how much of different products are in that cellular system at that exact time. So it's just more important to remember majority of ATP is produced in the electron transport chain during cellular respiration. It could be 32, it could be 34, 36, 38. A range of about 30 to 38 ATP comes from the electron transport chain, while only about 2 molecules of ATP are coming from the Krebs cycle, and about 2 molecules of ATP are coming from glycolysis. There is a really good video that I've posted for you guys that is going to help summarize this information. So I want you guys to look for the Amoeba Sisters cellular respiration video. That is a great way to follow this whole process from beginning to end. They've got visuals that really help it stick. And then there's also going to be a video available to you that summarizes redox reactions. And I feel like it's really helpful because that's a theme that's going to keep on coming back as you go through different science classes. So it's really good to understand exactly what's happening in those redox reactions and how they feed into each other. If you have any questions, always feel free to ask me and I'll be happy to help. Other than that, we made it through chapter 7. Now you understand cellular respiration. Feel free to rewatch this video as many times as you need. Pause it, take notes, do whatever you got to do to make it stick. Thanks, guys.